Hi, this is Miss Litton, and this is 7th period support for APBio. Say hi.
Hi.
Thank you. This is our last formal review. Okay, what we have left as far as reviews before I test on Friday is tomorrow. Wednesday is open for AP. So you can come in and ask me any questions that you'd like to have. If there's a group of you, say, hey, can you go over this again? Happy to go over it again. All the other reviews are out there. The only thing we have left to discuss is a very small portion in chapter 42, or section in 42. I'm going to take it back, not a small portion. We have 42 left to do, and we have the very end of chapter 20 to discuss, yes? And that's primarily about bacteria, which we have already discussed. Okay, so in my books, everything is done. I will tell you, just because you're going to get some big content on chapter 42 on Wednesday and then your test is Friday, I did not ask you an essay question on anything on Wednesday, okay? Because I thought that would be a fair trade if you've been preparing as you go along. Then it's not like you're sweating it out Wednesday night, Thursday night, trying to come up and write an essay. But what you should do is put a pin in it. Because if I don't ask you about it now, when would be a good time for me to ask you? Right, and I will not give that hint again. Okay, so make note of that to yourself, all right? As far as the essays, I will start on chapter 20, I promise. But this being our last formal review, the last couple of reviews, I've given potential essay questions during the reviews. I literally had those out and added others from ones I think we hadn't talked about. I think I added to it. Maybe not, because I think you had them all. I literally chose from those for your exam. So people took notes on it. I had 2 people email me the list because I didn't keep a list, and I said, hey, I told you what to get ready for. Email it to me and I'll use it when I write your exam. And I did exactly that when I wrote it, and I wrote your essay exams last night, all 3 versions. So pay attention to the list from the previous review and take advantage of those, all right? So on the big ticket items, your chapter 12, as far as that goes, as you're preparing for that, that's the molecular structure of the gene. So that one is all about the structure of DNA, right? How DNA replicates itself and the enzymes used in that. It's the structure of RNA, the process of transcription, right? The process of translation. You should be able to give me any of those steps in replication, transcription, and translation. You should be able to tell me every enzyme, every step. If I said, hey, that's about 5 steps, that's about 6 steps, give me the 6 steps, the enzymes, what happens, you should be able to do that. Okay, all of them are about 5 or 6 steps long. Okay, so be ready to do that on that, to be able to differentiate between DNA and RNA. Okay, so that was the how it works. Chapter 12. Chapter 13, we leveled up and said, okay, if that's how it works, how is it regulated? And in that process, we started small. We started with prokaryotes, and we looked at examples in prokaryotes. We talked about things like an operon and the parts of the operon. We talked about inducible and repressible operons, and we compared and contrast those. And you already wrote those down as your possible essays. We talked about prokaryotic versus eukaryotic control mechanisms and comparing and contrasting that. Inducible, repressible, positive, negative, prokaryote, eukaryote. Those are all things we went back and forth on. And we had examples, authentic examples for each one, right? And then I think we ended that one with talking about things can go poorly in that process, right? So we talked about different types of mutations, and we talked about cancer and, revisited those. So those kind of things could all come up in multiple choice as well as essay, right? Then chapter 14, we transitioned into biotechnology. And from that, we had an introduction to cloning. We talked about the 2 big techniques in recombinant DNA technology. We talked about the 2 different enzymes in order to make recombinant DNA. We talked about the polymerase chain reaction, and we talked about the value of using those 2, if you recall. And then we had examples. You had examples of bacteria, plants, and animals, and recombinant DNA technology being used in those, and for what purpose. And we compared and contrasted a little bit our nervousness at GMOs, and then we looked at an example where genetically modified organism was used in order to prevent, hopefully, a future famine with plants having some sort of pathogen, and how to protect from that pathogen when we looked at the spuds in Ireland. Then we looked at some more current ideas in the paradigm shift on the central dogma, because before it was always DNA, RNA, protein, as if the end goal was the protein. And now we're saying the end goal might be some copy of RNA, because it might be regulatory in some way. We looked at the idea of genomics, studying the full complement of the genome, not just the genes, but the intragenic and intergenic, right, parts of our genome. We talked about proteomics and studying proteins. Talked about the human genome. I know I'm skipping something. I can't remember what it is. And we talked about CRISPR, right? And we talked about the CRISPR complex and the two components of those and what each of those do. Right? So that is where we have been. Okay? Then our latest discussion is where we are now, and that's chapter 20. And in chapter 20, the big thing we want to start out with is just differentiating why viruses are not alive, right? And then we started talking about how they are not cellular, right? And then we talked about how if you want to study them, you actually have to put them in a living cell, because that's the only place they will replicate. And then we started talking about their origin. And maybe their origin was pre any type of cellular structures before the 3 domains, or maybe it was after, because they are host specific. We've got so much DNA moving around. Let's talk about some examples of DNA moving around. We could start with jumping genes, which are called what? Transposomes. So maybe it jumps out and becomes a virus at some point. There are small extra rings of DNA that are found in cells, and those are called, maybe that could be the beginning of a virus, or vice versa. We talked about exons and introns, and how we snip out the introns, right, and leave the exons. Maybe those are the beginning. Maybe there are little small nuclear RNAs, small interfering RNAs that become like a virus. But what we know about a virus is it definitely has some type of nucleic acid, and it definitely has some sort of what? Protein capsid, right? That nucleic acid is going to be one or the other, not both. It's either going to be what? RNA or, and then that RNA and DNA will either be single-stranded or double-stranded. And if it's single-stranded, it'll either be positive or negative, right? So there's all options on your nucleic acid, and then your protein capsid is going to give you your size and your shape around it. What else could you have in a virus? It doesn't have to have this, but what else could you have? An envelope. If it doesn't have an envelope, it's considered, I don't know, naked. Thank you. I was trying to act that one out, and I'm like, no. So it would be naked. But if it has an envelope, it's probably getting that envelope from where? The host cell that it just took over, right? And it may or may not contain what else? Makes reactions go faster. Enzymes, yeah. Remember, all the components of a virus, of every single virus, was made by its host, right? Because your nucleic acid that it has is just a code, and your cells know how to read code, they will code out a virus just as easily as they would code out some other enzyme that you need. That's why they're so vicious. And from that, we learned about some defense mechanisms that we harvested and talked about in chapter 14 that bacteria have against viruses because they don't want to be infected by a virus. What were those? Restriction enzymes or restriction endonucleases, which restrict the growth of an invading virus by cutting up its restriction enzyme. And what else did we learn about? CRISPR, right? And how that would also do the same thing, but it has a guide when it doesn't. All right? So then once we got that straight, then we started talking about just the basic, I don't want to say life cycle, but reproductive cycle. Why am I not saying life cycle? not alive, right? And how are we going to remember those steps? APBio, mister. So let's just say them. Let's look over here, not on the board, and see if we can say what each word stands for. What's number one? Attachment. Penetration. Got to get the nucleic acid in. Good. Making the components, viral components. Putting it together to form a full-fledged virus and release. And we know it could lyse the cell, or it could bud out and take a little bit of the envelope with it. If it lyses the cell's going to what? Just die. For sure die. So we learned the basic structure, and then we said, let's level it up. There's what you call the lytic cycle, which is what we just went over and learned. But you can take an off-ramp. When does the off-ramp, where does the off-ramp start? Following what stage? Penetration. Following penetration, we can take an off-ramp, and we can go into what? What's the stage? Starts with an I. Yeah, off-ramp starts with a vowel, and so does integration also starts with a vowel. I'm just going to say that. Okay, so you are going, opportunistic infections, okay. Anyway, integration. So during integration stage, you now enter the lysogenic cycle. Okay, lysogenic cycle, because you're lysing the what? Not the cells, the DNA, the genes. You're lysing the genes and integrating it in. Now, this is very stealthy. What are you called when you do this? You're laying latent, but what else is it called? What kind of phage is it called? Prophage. A bacteriophage is just one that only infects what? Bacteria. Yeah, a prophage or a provirus is one that has its genome integrated into the host genome. Now, two things from that. Number one, every single time the host genome gets replicated, when you make more cells, you're also replicating it with the viral genome intact, right? So every cell will have a copy of that virus in it. And at any point, it can be triggered and leave the host genome and then start to be re-enter to the lytic cycle. And where will it re-enter at? Biosynthesis, which makes sense, right? It went out following what? Penetration. Normally right after penetration is what? So it just goes off and back in, right? In at biosynthesis, and then we'll make more. Okay? All right, so then we looked at a better detail of that, which was here. And then I gave you some strong hints, right, about what I would expect on the test, to be able to diagram this, right, to identify the stages. Your diagrams would not be sloppy, neatly identifying that it matches the description for each stage. You remember all the key terms that we discussed, right? Do not miss that one if that shows up on your test, okay? Then we leveled up and we looked at an example of an animal virus. And how was this one different? Is it lysing the cell? No. Okay? So this is one that has an envelope, right? We had not seen one with an envelope yet. But you'll notice it gains access, right, via receptors that we have. It knows our code, and it gets right on into our cells. Our cells immediately take off its coat, take off the capsid, expose its nucleic acid, and we're like, what is your bidding master? And we begin to then help it to enable to make more of itself. Now, this virus, as is not going in and becoming a prophage, but it could. Okay, this one is budding off, which is typical, but it could also lyse when you have an animal virus. Sometimes there are naked viruses, right, and some of those ones with coats, so I don't want you to think that's the only way it could do it. Okay, then we leveled up one more time, and we talked about retroviruses. And retroviruses come packaged with, here you can see two different enzymes, right? What's one key enzyme all retroviruses have to have right away? Reverse transcriptase. And you're going to use that to get a what? A DNA copy of the viral RNA. And then you got to have it double-stranded. Then you need the enzyme what? Integrase to get it into the host cell chromosome, right? And then once it's in there, again, it can be a prophage, provirus. It can lay latent until it's ready to come out. And when it comes out, all the same things are going to happen all over again that we've already talked about. Okay, so that is a retrovirus. And then the next thing we did, we talked about how bad is it? And so we use two words, pathogenicity and virulence. And which one was the measurement of the other? Virulence. Yeah, so it's how bad is it as a pathogen. Pathogenicity says, is it a pathogen? And virulence is, how bad is that pathogen? And that's a measurement. We also address the fact that as we have climate change, it's going to change the environment, and it's going to change, and then organisms that maybe couldn't live in a place now will be able to, and with them will migrate all the diseases associated with that particular environment. Okay, then we said, but yet there is more that can cause danger and trouble. Now, viruses are composed of two things. What are the two things that viruses are composed of? Protein and nucleic acids. So, if you just have a naked piece of RNA, it's called a viroid. It's not a virus, right? Because it doesn't have the protein capsid. It's just a strand of RNA, and it can cause problems, especially with crops. So this is without, and that is with. Okay. Then we said, okay, virus composed of two things. What are the two things? Nucleic acid and proteins. So you can also have rogue proteins. Misshaped proteins, which trigger other proteins to become misshaped, and then you could have things like mad cow disease or something along those lines. Same kind of symptoms, right? Then we left our viruses and we moved into bacteria. And one of the first things we talked about was Leeuwenhoek and the development of the microscope, and having even awareness of small things. We talked about disproving spontaneous generation, that people used to think that living things could come from non-living things, like fungus or mold could grow out of an old soup pot, and frogs could come out of the mud, and rats could come out of the trash. So Pasteur, the cool thing you want to always associate, I know you will associate pasteurized milk, but Pasteur, you want to remember that swan neck flask, because that enabled the air to still go in to allow for aerobic organisms that were developing in the soup. They still had access to air, but those spores or microorganisms or bacteria would get trapped in the bottom of that swan neck flask and not infiltrate that fluid. Another man who did something similar with meat was named Redi, R-E-D-I, disproving spontaneous generation. Then the next part was just reviewing the parts of the prokaryote. Okay, I don't envision myself asking you an essay this unit on the parts of that, because I have a limited amount of time that I have to ask you questions, and I don't want to revisit something I've already asked you about. So I wouldn't stress about that part of it. Okay, I'll let you know as we finish up the parts of this prokaryote, what are the key things I would know for the essay as we finish it, because none of you got to the one part that I would consider putting in, which is really simple. I mean, like 4th grade simple. Okay, do you want to know? Yeah, it would be all the different types of symbiotic relationships, and there are only 3. And if I was going to tell you right now, and I'd want you to know an authentic example of each, let me get the simplified picture. It's right here. Okay. So there's 3 kinds of there are more than this. I'm only focusing on 3. It's always good for somebody in the relationship. It's always good for somebody. Okay? So mutualism is where it's good for both. Now this relationship of which we speak, bless you, has to be 2 different species, right? Has to be 2 different species, like you and your dog, if you have a dog. Okay? It's not like between you and your parents or you and your teachers. It has to be 2 different species. So mutualism is good for both. Commensalism, it's good for one, doesn't affect the other one at all. And parasitism, good for one, and it's bad for others. And then we'll talk about some authentic examples in class tomorrow. But I'm sure you could find some, but we'll look at authentic examples, and so you should be able to recognize that and speak about that. Just to practice it, could you answer each scenario 1, 2, and 3, what type of relationship each one is? And then why is this a good model and why is this a bad model? Okay, so what do you think this one is? Mutualism, because it's good for both. What about this one? Probably commensalism, because this one looks like parasitism. Why is this a poor model? Same species. Good. That's what I wanted you to recognize there. Okay, do you have any questions that you would like to ask me today? I think this is a test that you can have a strong showing on. I really do. I think it's material that you can master and know. It's going to be one of those ones where you either know it or you don't know it. Because it's spelled out for you what the expectation is. You're either going to meet that expectation or you're not. I would choose meet. Okay? Right? Okay, any other questions or concerns? Okay, I love you and I'll see you tomorrow.